As we leave this particular module, our last movie is going to be discussing protein structures. Now, of course, to just have blithely one movie on protein structures it's not just superficial, it's not just scratching the surface, it's kind of weaving the movie over the surface, if you know the reference for how to make a good martini. So all I'm going to do here is I'm just going to throw some facts and some pretty pictures at you in the hope that something will stick a little bit, so when you get into a protein module in your university studies, well then at least you'll have a teeny little bit of foundation for it. Anyway, we describe proteins, which are of course just large polypeptides, via 4 levels of structure. It's the so-called primary structure, which is where we just describe the amino acid sequence. Okay, so here you've got an example where you've got a histidine to an aniline to a valine to a leucine to a glycine to another glycine to a lysine to a lysine. Okay, in this teeny little part of the peptide strand. Okay, now of course those are held together by covalent bonds. Now at this point I need to make a little disclaimer. I'm using a picture here that you can see, just the first part of it, that I took from the internet. However, I could not find any reference to the original source of this picture. If you put in protein structure types or order or something like that in your Google image search, well then you'll hit this particular figure I'm about to show you is in 15 or 20 of the first 40 or 50 hits you get. Everyone uses it because it's a really good picture, but nobody actually said where they got it from. So I can't say where I got it from other than the internet, because none of the other sneaky people before me said where it came from. So if by some teensy little chance the original author or artist of this amazing picture happens to see my silly little movie, please email me, tell me who you are, and I will give you the appropriate credit, because it is really a lovely picture. Anyway, so protein, polypeptide, okay, those amino acid residues just all arranged nicely in a nice order, and that is the primary structure of the protein. Well, then there's the secondary structure, which is how parts of that long peptide chain are going to arrange themselves, okay, described as the 3D structure of the peptide backbone. And so you can see how in this particular case there's the amino acid residues kind of curling round in a nice little alpha helix, as it's called. And the secondary structure, okay, is held together pretty much by hydrogen bonds. Now there's two big types of secondary structure that is worth mentioning. First is the one I showed you there, the alpha helix. Okay, and remember what we're going to have is we're going to have different amino acid residues in this alpha helix that aren't next to each other at all, but are just part of the helix. And as the helix is formed, these amino acid residues get close enough to each other to hydrogen bond. So here is my attempt at an alpha helix. So we're going to start up here at the N-terminal end, okay? And so this is an amino acid, and I don't really care what the R group is, but I have identified it with a little orange ball. So here's the first residue, and then we've got its carbon, double bond oxygen, and then we've got a peptide link into the second residue. There's the nitrogen in that second residue. Don't care what the R is, but I've given it a blue ball. Moving on to its carbon oxygen bond, there's the peptide, and then moving into the third residue, the fourth residue, the fifth residue, the sixth residue, the seventh residue, the eighth residue, and the ninth residue. Now, as I hope you notice, as this curls around, okay, we're getting amino acids from one part of the chain hydrogen bonding with amino acids in a later part of the chain, and it's those hydrogen bonds that are keeping it in this little helical cycle. Okay, now you should notice that all the R groups are on the outside of this little spiral, and that's extremely important in terms of putting together later aspects of the structure. Now the hydrogen bonding, and I've tried to get it as plain as I can in this picture, it's between every fourth residue. Okay, so if, for example, here is an orange residue, that's a residue, right? And there's its CO, okay? And I've got the oxygen of that carbonyl group hydrogen bonding through the NH of another residue down here that I've also colored in orange, okay? Now between those, you've got 1, 2, 3, so that's the fourth residue away. Now this middle orange residue, okay, there sits NH that's hydrogen bonding to the previous orange residue. Here sits carbonyl that's hydrogen bonding to another orange residue. That is 1, 2, 3, 4 residues further on, okay? So I've got the orange is the first, fifth and ninth residues. The blues, right, are the second, third, fourth, fifth, sixth residues. So again, 4 separating them, and there's the blues carbonyl going to the next blues NH, okay? And the same relationship between the greens and the purples, okay? So hydrogen bonding every fourth residue, and of course that keeps it in a nice repeatable nice symmetric structure. So symmetric that there are 3.6 amino acid residues per turn, okay? And that's why you don't see these orange ones matching up exactly, because there's not 4 residues per turn, there's only 3.6 residues per turn, okay? And just for information's sake, that's 540 picometers, right, per turn, okay? So as we go from one turn all the way through to sort of round here would be where the turn starts again, okay, 3.6 amino acid residues away, that's 540 picometers. Now that 540 picometers means absolutely nothing to you right now, but in a minute we'll compare it to the distances associated with repeating units or repeating parts of the other big type of secondary structure. Now there's obviously any amino acid can take part in any type of the structure, but what's found as people have analyzed hundreds and hundreds and hundreds of proteins is there are little patterns to them, okay? By and large, alanine, cysteine, leucine, methionine, glutamic acid, glutamine, histamine and lysine are more likely to be part of alpha helices than they are other types of secondary structure. And what one also finds is that if there are too many bulky R groups in an alpha helix, it's not good. They get in the way of each other, okay? And if you match these up, these are by and large the smaller of the amino acids. Obviously glycine, the smallest, isn't there, but they are the smaller of the amino acids. And another thing that's noticed is that if you have ionic amino acids, those are the amines or the acids that when you're at neutral pH are in their charged conjugate forms, you can have them as part of the alpha helix, but if you have too many of them close to each other, then that's not good for the structure at all. So in other words, when you look at alpha helices in proteins, you find not a whole lot of bulky R groups, and the ionic ones in there, the glutamic acid and the lysine and histidines, which can be and are charged at neutral pH, they tend to not be close to each other. Now the alpha helix is one type of secondary structure. So beta sheet is the second major type, in which peptide chains can orient themselves and interact with themselves in a sort of a local basis. Okay, now if we think of here is a chain of amino acid residues, so here's the first residue, the amide group, second residue, amide link, third group, amide link, fourth amide link, and so on. Okay. Now, what I want you to notice is how these are arranged so that you've got the R groups are on alternating sides of the chain. Here's the R group coming out at you, the R group going away, R coming out, R going away, R coming out, R going away. And of course, the R groups will do that so that they're minimizing the steric sort of disruption and the interference between one group and another group. Now, there's two ways in which we can get a second part of the long peptide chain in the protein orientated with respect to this one. First way is what's called the parallel arrangement, and here is the first part of the chain, and then here's a later part of the chain that is oriented in a way that it exactly matches the first one. Indeed, all I did was I took this chain and I copied it to make that chain. So you can see we've got the C double bond O's pointing up there, and then the NH down, C O's down and the NH up. Next residue, CO up, NH down, as we go all the way through there. Okay, now this chain and this chain can of course hydrogen bond with each other quite nicely with alternating NHs hydrogen bonding with COs. So there we are, there's some hydrogen bonds right there. Okay, so we got the NH at the top with the CO of the bottom, and then a bit further on the CO of the same residue here with the NH of a different residue on the next one, indeed 2 residues away from the next one, and so on and so on and so on. And then we got the CO of the same residue on the bottom with the NH of a residue 1 away from the first residue on the top. Okay, so you get this nice little parallel pattern here. The other way that it can range is like this. Now this is exactly, this second chain is exactly this first chain, except what I've done is I've had to rotate it a little by 180 degrees. Okay, so now you see we've got one residue here, one residue there, flipped around, okay, reorientated, and so on and so on and so on. And indeed, you can actually have a really easy clue as to what I did, because here is the C end of this cut off there, there's the C end left of that one there. So just took this picture, rotated it, could get this so-called anti-parallel arrangement. Now the anti-parallel arrangement actually has a lot more hydrogen bonding within this picture here, because instead of it being a sort of little staggered arrangement, the NHs and the O's are exactly right above each other. Okay, so parallel beta sheets, anti-parallel beta sheets. Told you to remember the 540 picometers from the alpha helix repeating pattern. There's a similar distance between identical residues in the parallel and the anti-parallel, 650 picometers and 750 picometers between the distance between where it starts to repeat itself again. Okay, so beta sheets, obviously a nice sort of sheet arrangement, and of course I could have gone on and on and on with another row under here, another row under here, and so on to give you that sheet arrangement. Now in the same way that the smaller amino acids were found more often than not in alpha helices, the slightly larger ones, valine, isoleucine, there's phenylalanine, tyrosine, tryptophan, the bigger amino acids are found associated with beta sheets. Now what I'm not drawing here is of course that there are loops that you can get. So one beta sheet, obviously you've got the peptide linkage here where it's going to go on and go on and go on, or sorry, the peptide chain here is going to go on and on and on. Well, at some point it's got to loop back here so that you can have this second part of the chain interacting with this first one. Well, those loops are really rather fascinating. That's where glycines will come in. That's where the cis arrangement of proline will be associated, and that's where some of the other amino acids that are missing from my first 2 lists are often found, in those sort of loops that change the directions or that connect alpha helices to beta sheets within the big protein.